When you think about anti-aging or, tissue repair, you probably picture it a lot like architecture or masonry.
Yeah, like structural work.
Exactly. Like we freeze the scaffolding so it doesn't shift around. We inject synthetic fillers to kind of spackle over the cracks and just, take up empty space under the skin.
Right, which is essentially preserving a facade. I mean, you're addressing the visual symptom of the aging or maybe the physical void left by tissue damage, but you aren't doing anything to change the underlying biological behavior of the tissue itself. It's a completely static intervention.
But imagine if, instead of just propping up a crumbling wall, you could actually hand the cellular construction workers a highly specific blueprint, drop off the exact raw materials they need, and tell them to, rebuild the foundation from scratch.
That's the dream, right?
Right. So welcome to this custom-tailored deep dive. Today we are looking at exactly that. We're exploring how polynucleotides signal cellular repair. And I have to say, the stack of material you provided for us to sift through today, it is wonderfully eclectic.
It really is all over the place in the best way.
Oh, absolutely. I mean, we are pulling from dense, older textbooks on genetic replication, agricultural patents, complex clinical data on menopausal tissue repair, oncology research, and even cutting-edge guides from aesthetic clinics.
Yeah, it definitely seems like a disjointed stack of reading at first glance. But, there is this brilliant through line holding it all together. Understanding the mechanics behind this specific type of molecular communication, it completely alters how we view tissue regeneration, regardless of the medical discipline. Knowledge is always most valuable when you can see how broadly it applies.
Right, because when people hear terms like tissue regeneration today, they immediately think of freezing muscles with Botox or pumping in hyaluronic acid.
Which isn't true regeneration.
Exactly. But we are looking at literal genetic recycling. We're talking about using the universal alphabet of DNA to instruct human cells to act young and heal.
Yes, the universal alphabet.
And before we get into the practical application of how we're actually using these molecules for glowing skin or severe wound healing, we need to establish what these molecules actually are at a base level.
The foundational blocks.
Right. So looking at the older view of genetics text from your stack, it brings us right back to the Watson and Crick model of the double helix. We are talking about the basic AGCT alphabet. So that's adenine, guanine, cytosine, and thymine.
Yeah, and those four bases, they are the foundational genetic instructions for all known life.
Literally everything.
Everything. So when we discuss polynucleotides in a medical or an aesthetic context, we are simply talking about long, isolated chains of those specific nucleotide monomers connected by phosphodiester bonds.
Okay.
It is the core structural backbone of DNA.
And because this AGCT alphabet is identical in everything that grows, it means this signaling language isn't just a human phenomenon.
No, not at all.
Which explains why one of the patents we're looking at in your stack is actually agricultural.
That was fascinating.
Right. They are using exogenous, meaning, from outside the organism, polynucleotides to increase plant yield and biomass. The patent details how they're integrating these nucleotide chains directly into plant chloroplasts to boost photosynthetic efficiency.
Yeah.
And then, right next to that in the research pile, we have a patent targeting specific polynucleotide sequences for human cancer treatments.
Yeah, and let me contextualize those cancer targets, because I think it really highlights the versatility of this mechanism.
Go for it.
So the patent specifically mentions sequences like FM26F and MGC2498.
Catchy names.
Very catchy, right? But these are particular genetic sequences closely associated with abnormal immune responses and the tumor microenvironment.
Okay.
By introducing specific exogenous polynucleotides, the goal is to modulate how the immune system recognizes and attacks those cancer cells. You're essentially using the universal language of genetics to rewrite a faulty command. The cellular machinery knows exactly what to do with these molecules, whether it's, a leaf trying to capture more sunlight or a human immune system trying to flag a tumor.
That's wild. I was trying to visualize this process while I was prepping, and I keep coming back to a Lego analogy.
Okay, let's hear it.
So think of polynucleotides like taking a massive, destroyed Lego castle. You break it down into individual, perfectly intact bricks, and then you hand those raw bricks directly to a master builder, which is your cell.
Right.
You aren't just giving them random material. You are giving them the exact pieces they already know how to use to build a new structure.
Yeah, the Lego analogy works on a surface level, but the biology is actually a lot more dynamic than that.
Okay, how so?
Well, you aren't just handing the cell bricks. You're handing them bricks that actively communicate with the site manager.
Oh, interesting.
You're providing the substrate, yes, but the real breakthrough is how the cell interprets the arrival of those materials.
Right.
The literature highlights two very specific biological pathways that make this repair possible, the first being the salvage pathway.
Okay, so let's unpack this. I was reading the clinical breakdown of how this happens, and it mentions that the salvage pathway bypasses something called de novo synthesis, but honestly, I couldn't quite wrap my head around the mechanics of how that actually saves the cell energy during a crisis.
Yeah, it's a bit dense. Let me break that down.
Please.
Normally, if a cell wants to repair its DNA, say after you've been sitting out in the sun and sustained UV damage, it has to build new DNA from scratch.
Okay.
That process is called de novo synthesis. You can think of it like trying to build a house by mining iron ore and forging your own nails.
Oh, wow. That sounds exhausting.
It is biologically exhausting. If the tissue is already injured or hypoxic, meaning it's starved of oxygen, it just doesn't have the metabolic energy to do that heavy lifting.
So it just gives up.
Yeah.
And the tissue ages or dies.
Often, yes. But when we introduce exogenous polynucleotide fragments into that damaged tissue, natural enzymes in your body called nucleuses immediately recognize them and start cleaving them.
Like chopping them up.
Exactly. They chop these long chains down into individual, ready-to-use nucleotides.
Okay.
The cell then recycles these raw materials via the salvage pathway. It skips the energy-intensive mining and forging process entirely and just uses the bucket of nails you've provided to repair its own DNA.
Wait, so the physical breakdown of the product is actually the treatment itself.
Yes.
Because in traditional aesthetic medicine, patients are used to a filler being injected, and as the body naturally breaks it down, the treatment is like wearing off.
Right, they see the breakdown as a negative.
But here, with polynucleotides, the breakdown of the chain by your enzymes is exactly what kicks off the healing cascade.
That is the crucial paradigm shift.
Wow.
But the salvage pathway is only half of the story. The second mechanism is the A2A receptor-mediated pathway.
And the A2A pathway, what's that do?
As those polynucleotide fragments are chopped up by your nucleuses, one of the resulting byproducts is a molecule called adenosine. This adenosine naturally binds to the A2A receptors sitting on the surface of your cells, particularly fibroblasts.
And those are the ones making the good stuff.
Right, exactly. Fibroblasts are the specialized cells responsible for churning out collagen and elastin.
Okay, so when that adenosine docks into that receptor, what happens next? Because the research mentions a massive biological cascade.
It really is massive.
It starts with the release of CAMP, or cyclic adenosine monophosphate, which acts like a cellular messenger. But then it drops into some heavy terminology, noting that it suppresses the NF-κB and MAPK pathways.
Yeah, it's a lot of acronyms. Think of the NF-κB and MAPK pathways as the cell's master alarm switches.
Okay.
When tissue is damaged, these pathways flip on and trigger the release of pro-inflammatory cytokines like IL-6.
So they're the ones blaring the cellular sirens and causing the inflammation.
Precisely. And in the medical community, we spend a lot of time studying inflammaging.
Inflammatory.
Yeah, it's this chronic, low-grade inflammation that essentially acts like static noise. It prevents your cells from hearing repair signals, which accelerates the aging process.
Oh, that makes so much sense.
Right. So when adenosine binds to that A2A receptor, it shuts off the siren. It suppresses those alarm pathways.
Wow.
Switching off that chronic inflammatory signal while simultaneously giving the fibroblasts the green light to build new collagen, it's a masterstroke of biological engineering.
And the cascade doesn't stop there either. That same A2A receptor activation upregulates VEGF. That stands for vascular endothelial growth factor.
A very important one.
Right, which promotes angiogenesis, the creation of brand new blood vessels to deliver vital oxygen back to the tissue.
Yes.
And on top of all of that, it increases the body's own natural hyaluronic acid synthesis by up to 40%.
Incredible, isn't it?
It is. No synthetic gel fillers, just your body remembering how to make its own moisture. But all of this incredible biology begs a very weird practical question.
I think I know where you're going with this.
If we need this highly specific biological language to trigger the A2A receptors, where is the raw material actually coming from?
Well, the clinical guides and patents indicate that the polynucleotides used in these medical treatments are sourced primarily from the gonads and sperm of chum salmon and rainbow trout.
Okay, I have to pause and speak for the listener right now. Are you telling me that we are essentially taking sterilized fish sperm and injecting it into our faces and bodies?
I completely understand the reaction. It sounds bizarre on its face.
It really does.
But from a biochemical standpoint, we have to look at biological efficiency. Why fish and why that specific reproductive tissue?
Okay, why?
During the spawning phase, a salmon's physiological energy is almost entirely redirected toward reproduction. The gonadal tissues become incredibly dense, packed with sperm cells containing highly condensed DNA.
Right.
In fact, DNA makes up to 7.5% of the entire wet weight of that tissue. It is simply the most concentrated, efficient source of raw nucleotide material available in nature.
And the data points out that salmon DNA has over 90% homology, or, similarity, with human DNA protein coding sequences, which goes right back to our earlier point about the universal alphabet of life.
Yeah.
But obviously, they don't just extract this and, put it straight in a syringe.
No, certainly not. The extraction undergoes a rigorous purification and sterilization process at 121 degrees Celsius.
Okay.
This high-temperature moist heat treatment systematically removes all proteins, peptides, and lipids from the raw material.
Which is important because...
Because what you are left with is a highly pure, non-antigenic substance. The proteins are what typically cause allergic reactions, right? So because they are completely removed, the immune system doesn't flag it as a foreign invader.
Oh, that makes sense.
Though I should add, the clinical guidelines do note that it is not recommended for vegans or those with severe fish allergies.
Just to be safe.
Primarily out of an abundance of caution, yes.
So looking at the manufacturing data, there's another key detail called the Marquis polynucleotide cutoff. They categorize this extracted DNA based on molecular weight, specifically whether the chains are over or under 1,500 kilodaltons.
Yes, the Marquis cutoff.
For someone trying to visualize that, what does 1,500 kilodaltons actually represent in terms of how it acts in the body?
To put 1,500 kilodaltons into perspective, imagine we are separating the massive, heavy cargo ships from the tiny, agile speedboats.
Okay, like this.
If the chain is over 1,500 kilodaltons, it's classified as a polynucleotide, or PN. These are the cargo ships.
Got it.
They are larger, longer chains that degrade very slowly, acting as a structural and hydrating scaffold in the tissue to provide sustained long-term stimulation.
So they hang around for a while.
Exactly. Now, if it's under 1,500 kilodaltons, it's called PDRN. These are the speedboats. They are shorter, lighter chains that can quickly penetrate compromised tissue to do rapid, targeted repair work.
So the speedboats get to work immediately on a specific crisis, while the cargo ships stick around to hold up the harbor walls.
Precisely. The molecular weight dictates the clinical application.
Which brings us to the real-world impact. The molecular theory is fascinating, but how is this cellular signaling actually being utilized in hospitals and clinics today? Because the sources we are looking at cover a shockingly wide range of applications.
It's remarkably broad.
Let's look at the wound healing data first, because it proves this isn't some brand new cosmetic fad.
Oh, definitely not. PDRN has a robust 30-year history of use in hospital wards. The clinical trial data on diabetic foot ulcers is particularly compelling.
Right.
These ulcers are notoriously difficult to heal because the patient's microvascular environment is heavily compromised. The capillaries are destroyed, leaving the tissue hypoxic and starving.
But then they use the PDRN.
Yes. By administering 5.625 milligram doses of PDRN to these patients, they actively utilize the energy-saving salvage pathway and the blood vessel-building properties of the ATA receptor.
And the results were amazing.
Staggering. They almost doubled the rate of complete wound healing compared to a placebo.
When you think about what that means for a diabetic patient who is staring down the barrel of a potential amputation, it is profound.
Life-changing.
And then you take that exact same biological mechanism and move it into a high-end aesthetic clinic. The cosmetic dermatology guides show practitioners using it for under-eye bags because that specific skin is so thin and delicate that traditional fillers often look lumpy or draw in too much water, causing puffiness.
Yeah, HA fillers there can be tricky.
Exactly.
Yeah.
They're also using it for scarring alopecia to quiet the scalp inflammation and allow hair follicles to regenerate. They even use it to treat severe erythema, the deep redness you get after aggressive laser treatments. It actually signals the skin to replace disorganized, stiff, scar-type collagen with healthy, flexible structural collagen.
It really just proves a fundamental truth about our biology, which is that the mechanism of cellular regeneration is blind to the aesthetic or medical context.
What do you mean by blind?
Well, if you give the body the raw materials and the precise signals it needs, the specific location of the tissue doesn't matter.
Right.
The biology of repairing a diabetic ulcer on a foot is remarkably similar to the biology of repairing UV-damaged, thinning skin under the eye.
And that universality extends to intimate health, which might be one of the most impactful applications in this entire stack of research.
I agree completely.
There are several detailed clinical studies here focusing on genitourinary syndrome of menopause, or GSM. As estrogen levels drop during menopause, women can experience severe vaginal atrophy, extreme dryness, and dyspareunia, which is incredibly painful sex.
Yes. Historically, the first-line treatment for GSM has been local estrogen therapy to artificially replenish what the body has stopped making.
But that isn't a viable option for everyone, right?
Not at all. Women who are breast cancer survivors, for instance, often have strict medical contraindications for estrogen therapy.
So they just suffer.
For those patients, GSM can severely impact their psychological well-being and relationships, and for decades, their treatment options have been largely limited to temporary synthetic lubricants.
Enter polynucleotides. The studies detailed treatments using polynucleotide ovules or intradermal injections with products like nThe that completely bypass the hormonal system.
Which is huge.
Massive.
Yeah.
By delivering these specific DNA fragments directly into the vulvovaginal tissue, they stimulate the local fibroblasts to build new, healthy collagen. They increase natural lubrication by boosting the body's own hyaluronic acid production.
Yep.
And they dramatically turn down that master alarm switch to reduce the chronic inflammation causing the pain.
It's incredible.
Looking at the trial data, one study noted that 100% of the participating patients reported significant improvement in at least one domain, whether it was a reduction in itching, improved natural moisture, or a decrease in sexual discomfort.
It is a profound application of regenerative medicine. You are not artificially lubricating the tissue. You are instructing the cells to regain the functional, structural capacity they had years prior. But, as with any medical intervention that relies on coaxing the body's natural biological processes, there are realities and limitations that patients must understand before undergoing treatment.
Yeah, right, because reading through this, it sounds like a miracle cure. But biological repair takes time. The clinical guidelines are very clear that this is not an instant lunch break fix.
Not at all.
Achieving these baseline results requires 3 to 4 distinct treatment sessions, usually spaced a few weeks apart.
And your results don't actually peak until 3 to 6 months later, as that new collagen network is slowly woven together.
Furthermore, the physical process of delivering these signals into the tissue isn't always comfortable. Delivering these molecules requires a mesotherapy technique.
Right, the multiple injections.
Yeah, which means dozens of tiny injections across the entire treatment area to ensure the signal is evenly distributed.
And the aesthetic guides are very upfront about the immediate physical realities of that technique.
Oh, they don't hold back.
The injections can sting. They cause what the industry calls blebs.
Blebs.
Yeah, blebs are these small, highly visible bumps of liquid sitting just beneath the skin's surface, almost like a grid of mosquito bites that can take several days to fully absorb into the tissue.
And if you are treating a highly vascular area, like around the mouth or the eyes, the risk of significant bruising is incredibly high.
Yes.
One clinic source literally wrote that this treatment is not for the faint-hearted. Which makes me want to ask you, the listener, would you rather endure a few days of looking bumpy and bruised to achieve genuine, permanent cellular repair, or would you stick to an instant but temporary fix that just masks the underlying problem?
That's the real trade-off. It ultimately comes down to a question of delayed gratification versus immediate reward. And there are genuine risks if the treatment isn't respected by the practitioner. Because polynucleotides are fundamentally different from traditional hyaluronic acid fillers, they cannot simply be dissolved with an enzyme if an amateur injector places them incorrectly.
Oh, wow. So you're stuck with it?
If injected too shallowly or in the wrong concentration, they can cause persistent lumps. Professional expertise and deep anatomical knowledge are non-negotiable here.
Good to know.
True homeostasis, bringing the damaged tissue back to a state of youthful, functional balance, it takes patience. You have to remember, the physical breakdown of the DNA chains by your body isn't the treatment wearing off. It is the treatment just beginning.
That is such a powerful way to reframe how we think about medicine and healing. Okay, let's recap the incredible journey we've taken through these sources.
It's been quite the ride.
Truly. We started with the foundational AGCT alphabet of genetics, discovered by Watson and Crick. We explored how the body uses the energy-saving salvage pathway to recycle those biological letters, bypassing the exhausting de novo synthesis process. We looked at how adenosine molecules lock into the A2A receptors to shut down the cellular alarms causing inflammaging, all while triggering new collagen and capillary growth. And we traced how highly purified, incredibly dense DNA extracted from spawning salmon is crossing species lines to do everything from rescuing aging facial skin to healing severe diabetic wounds to restoring intimate health and quality of life for breast cancer survivors.
It really is a profound testament to the shared underlying architecture of life on this planet.
Absolutely.
Which leaves us with a final, lingering thought to consider. If we can isolate this universal language of cellular repair, crossing species barriers to heal everything from a plant's chloroplast to a human being's compromised skin, what other ancient genetic instruction manuals are hiding in nature, just waiting for us to learn how to read them?
That is a brilliant question to end on and a perfect encapsulation of why we dig into this material. Thank you to the listener for sharing this incredibly diverse stack of sources with us, and thank you for joining us on this custom-tailored deep dive. We'll see you next time.